how are you doing? Welcome back to studying Prescott's Microbiology, and today we are going to talk about the cytoplasmic matrix and some of its structures, two of its structure. The first one is what is cytoplasmic matrix? And we are talking about the cytoplasmic matrix of the prokaryotic cells. The second thing is the prokaryotic. We have to know what the structures are, and remember the prokaryotic cell cytoplasmic matrices structure are less, right? Because they are not that complex. Like they usually have ribosomes, prokaryotic skeleton, and these skeleton, ribosomes, and inclusion bodies, right? The inclusion body number is high. Now we are going to discuss what the cytoskeleton is, what are the inclusion bodies, what are these things, right? Now I think people are familiar, those who study the eukaryotic cell, but anyway we are going to go into the detail, right? So the thing is, first let's know what the cytoplasmic matrix is, right? So we know, right, what is the cytoplasm is. So it's the same thing as cytoplasmic matrix, like the thing in the cell membrane, present in the cell membrane, which have the ribosomes, nucleoid, and other organelles. That water type, constituency type material is the cytoplasmic matrix. Okay, so now what is protoplasm then? So if you combine cytoplasm plus all of the organelles plus the plasma membrane, that makes it protoplasm, right? So we are talking about the cytoplasmic matrix. The cytoplasmic matrix is usually water. In bacteria's case, it is 70% of the water, right? And some here and there inorganic salts and minerals and things like that, right? I'm not talking about the organelles, okay? Organelles, nucleoid, these things are separate from the cytoplasmic matrix. So that 30% is the salt and minerals and things like that, right? I think it is not that much different. Well, of course it is different than eukaryote, but I mean the water quantity shows that it has a lot of water quantities. Now, what is the prokaryotic cytoskeleton? Now, first, generally, what is cyto? Cyto means cell. Skeleton is something that supports the structure, just as in your body, the skeleton. So cells have that skeleton as well, right? And especially because this is a single cell, so of course it needs to retain its structure, rather because it doesn't combine with others. Now, first, this was assumed that the prokaryotes didn't have the cytoskeleton. It's because we lacked the microscopy and we couldn't see that, right? Eukaryotes have very obvious structure that we call that, these are cytoskeleton and these support shapes, and they transport proteins and stuff like that. But prokaryotic cells didn't have that. So it is later that they found out that prokaryotic cells have cytoskeleton as well, and there are three types, exactly the same three types that eukaryotic cell has, but the naming is different and maybe the structure is different. You can say they are the counterpart of the eukaryotic cytoskeleton. So these are the three types. If you have tubulin, actin, and intermediate filament proteins in a eukaryotic cell, then you have FTSZ, MREB, and crescentin in the prokaryotic cell, right? So these are not controversial. These are the counterparts of each other. They work the same way, but their existence in different kinds of cell make them a little bit different, right? So now what? Before going towards the inclusion body, let me explain what these three are a little bit, or let me show the diagram with it, right? So this is FTSZ protein. If you look at it, it helps in the cell division, and it usually makes this ring-like structure to divide the cell. We know division occur in the bacterial cell, right? Then who allows the cell to divide equally? Who is it? So of course it is if this FTZ protein, or you can say tubulin in eukaryotic cell, and it helps in the cell division. Like look at this here. The cell that has ring formed gladly easily divided into two daughter cells of equal sizes and nothing different, right? But the one that didn't have this FTZ ring formed, look at the cell, it elongated even more, and the cell division arrested, means no cell division occurred in them. Now MREB, the second protein that acts as actin, it helps in the shape maintenance, and it is usually found in broad bacteria because it is in cylindrical shape, as is shown here. Not cylindrical, you can say spherical. No, not spherical. What do you call spring? Helical shape, right? So if you see it here, it is in helical shape and it protrudes outward. It forces the cell membrane to go outwards, like making it forcefully remain in the state that it is. So the one that has that always maintain the rod shape, right? Like look at this here. Look at this spherical. It doesn't have these MREB. If they had, they would have this as well. Of course, MREB and FTZ exist in the same cell, and the other thing is they can exist. All of three of them can exist together, right? So if you look at this, it only has the what do you call this? The spherical usually don't have that much cytoskeleton because it doesn't have that much of a shape, right? I mean, it has, but it don't have that. So it only has a ring shape. With a rod shape, you have the ring for cell division, and you have these helical type stuff. I mean, they showed it round, but it is not round. It is helical shape, and they help in rod shaping. Now you have the third one, and that is crescentin. Crescent. The name shows that it is in crescent shape. Crescent is like half C, right? The way you write it. So how does it make the cell curve? It makes the cell curve shape. So it is also in rod shape. If you look at this, I mean, you can't make the cell crescent, round cells crescent, because it will lose its spherical shape. So it is in rod shape. And how it does it, if you mark the line like this, the bacteria starts to grow from this areas. So the more the bacteria grows, the more curved it will be. Like, understand, right? Like the bacteria will elongate from these areas. Look at these red areas. It will elongate from these red areas to make it curved. So now that was that, okay? So generally that you have with a cytoskeleton, these three types, and again it's cell division, cell shape, and the protein transfer. Maybe like you have to transfer protein from one area of the cell to another, and because they are scattered all around the cell, like here if you look at this, all around the cell, so it is easier for them to do that work. Like here, I don't think if you can see the diagram clearly. Like look at this here. They have taken the microscopic picture, and if you look at these green, it is not exactly the MREB protein, but they combined it with a dye, green dye. It is a MREB like protein. It is not MREB exactly. Anyway, it acts the same, because look at this. So they enlarged the same picture. Here we can see. You can see the helical springy type things, right? These are MREBs, and that makes the shape rod shape, understand? So this is just the general idea of prokaryotic cytoskeletons. Like we need something to maintain our shape, our cell shape, and we need something to maintain that cell division, and for that we have cytoskeleton, and of course they do one few more things with that as well, right? If you don't have shape, you can't do functioning properly. You won't have enough space. You wouldn't survive because of your, what do you call, extra flexibility. So yeah, That was that. Now inclusion bodies. Now these are the bodies that are kind of vacuole like things, vesicles like things, granules like things. Like these are sacs, round and round, a membrane bounded area where you have different types of things in them, right? Now it can be the inclusion body. Wait, what does inclusion mean? Wait. Inclusion means, the action or state of including or being included in a group, within a group or structure, right? Yeah, it's a kind of grouping things, right? So now these inclusion bodies have organic material or inorganic material. It depends on what kind of inclusion body you have. Inclusion bodies are very high in number in bacteria, like everything that needs to be stored, every enzyme that needs to work separately, they put it in a membrane and made them work separately. Like you can say this is the low level of organelles. If you have high level of organelles in a eukaryotic cell, you have these kind of vesicles that work as a separate units, because they don't have that high level organelles, right? Now you will understand what I am saying as I explain it. So when I said it is of organic and it is inorganic material, it is as it states, because there are given example of the organic materials, like they store phosphate, they store carbon, they make carbon, right? Because they have enzymes for that. They also store glycogen. You know, right, that the prokaryotic's energy source is usually the glucose, right? So what will happen if you get rid of the glucose, right? Of course they would need that, so they store it in advance in a form of glycogen, just as we do in our liver. Now they also store phosphate, because there are also a lot of bacteria that depends on the phosphate working. So now also there are gas vacuoles that help the bacteria float. They don't necessarily do anything, that they just let them float, let them move, right? Specifically in water, like if they want to go deeper into the ocean, they will just pop out those gas vesicles, and of course they will be heavier and they will move towards down. And if they want to float up above as well, they will just round and make other vacuoles. Of course the gas vacuoles, they will only allow gas within, and of course they will float then. They float above for sunlight and oxygen, or you can say carbon dioxide, these kind of things. Well, I think they go downwards because for the nutrients and stuff like that. So the organic, of course you all know, right, what organic is, right? They have to explain. Alright, so organic inclusion bodies are for, like, the most common ones are the glycogen ones, or the poly beta hydroxyl alkanoids, right? Or for example, if you specify this alkane part, right, take a specific alkane, and that is butyrate, right? So we have that both the glycogen and the poly beta hydroxybutyrate is used for energy storage, right? In a compound like carbon molecules, they store that. Now if you look at the glycogen, of course it is organic, so it has carbon backbone, and it stores glucose unit. Like, it doesn't necessarily have to be the same glycogen that we store in our body. It can be different, but it will be glycogen because it will be in branch chain. Glucose are usually don't form. Of course glucose are in a form of units, but if you make the polymers of them and yeah, it makes the glycogen. Now the bonding is different. They have two types of bonding. The first one is the one like this one, A124, is linear, you can say, not branched chain glycosidic bonds. Like if you combine glucose in a linear form without branching them, these glycosidic bonds are this type, while the other one, they makes it branched chain. Like if you after you make a linear chain, and if you pop out branches of them, the chains, branch chains, more and more, and these are that kind, 1 to 6 alpha glycosidic bonds as well, because it starts from alpha, right? And then the number of carbon can vary, like the branch can be long and be short. So poly beta hydroxybutyrate, as you know that butyrate is not that long of an alkane, right? Like wait, alkane, methane, butane, no, or methane, ethane, butane, like it comes on a third number, right? So of course it is not that long, so it needs to make chains, right? Like poly for storage. Poly means many, so many of these beta hydroxybutyrate combines to make the poly one, right? Now how do they combine? If the above one is combined by the glycosidic bonds, this one are combined with ester bonds. We know what the ester bond is. You have O, which is attached to C on one side, and of course the regular normal C, while it is attached to C double bond O. Oh no, sorry, it is, yeah, am I wrong? What am I saying? Dang, I just went through it yesterday. Alright. I don't want it to go, to 2 carbon. Wait, what? Oh yeah, so of course I was right. So one O is attached to a regular carbon. Of course the R is alkane chain, maybe alkene carbon chain. So it is with the regular carbon, and the second carbon it is attached to have double bond O with it. So this is the ester bond. Now how do you create that kind of stuff? So if you have C double bond O, of course, so you have to make a bond between carboxyl and the hydroxyl group. If you make a reaction between them, they combine to form ester bond, right? And so I think the one side of the polyhydroxybutyrate has the hydroxyl group, while the other one has the carboxyl group. It is because it is 8. So they are formed by the ester bonds. Now these are just 2 organic inclusion bodies, right? Glycogen and the polybeta hydroxybutyrate. Both are used for the storing of carbon and the energy. Specifically, beta hydroxybutyrate is for the, when the cell is in the stationary phase. Stationary phase, right? Like when it is just getting accustomed to their environment and get used to their environment. At that time, they are waiting for certain nutrients to come so they can start to grow. Until those nutrients don't come, they want to store as much as of the nutrients that are still available. And they store those carbon types in a polybeta hydroxybutyrate state. Now, how do you look at it in the microscope? So they say that you color it with a Sudan black. Polybeta hydroxybutyrate are visible with a Sudan black. So when you do the Sudan black color, the solvents that we use, that dissolve the glycogen vesicles. So even you dissolve the glycogen vesicles, it disperses the whole through cell. Like if you look at this diagram, see small vesicles, and they are granules of the glycogen, right? It is because they are hydrophobic. Both the glycogen and the beta hydroxybutyrate granules are hydrophobic. So the solvents dissolve this one, while this one remains the PHBP, polybeta hydroxybutyrate. I don't have to pronounce it over and over again. So it is clearly visible under the microscope. Well, of course, unless you studied everything, you wouldn't know what this white structure looks like. But of course, these are microbiologists, or you can say scientists, that are studying them, so they know that this is the polybeta hydroxybutyrate. They do reactions with them, of course, the chemists, and so the other things. Cyanobacteria, if you look at it in a way, they have cyanophycin granules, and they store the nitrogen. And how do they store the nitrogen? They store in the form of amino acid like arginine and aspartic acids. They are high in number in them, and these bacteria of course are present in the bacteria that needs nitrogen in high quantity. Now the same, if you look at the cyanophycin granules that stores nitrogen, so of course there are inclusion bodies that of course deal with carbon dioxide. You don't know that bacteria are, so buddy with the carbon dioxide. So the carboxysomes fix carbon dioxide fixation. Not only does it fix the carbon, fixing carbon means that it takes the atmospheric carbon and convert it into organic carbon, like the glucose you eat, lipids, or things like that. Usually bacteria don't make that much complex things, like think about the plants, like when they do photosynthesis. So of course cyanobacteria of course do photosynthesis, so they do need carboxysomes. The enzyme that is used for the carbon dioxide fixation is the ribulose fixose. Now, unlike the previous three inclusion bodies that we studied, they store things, right? Like for example, nitrogen, carbon, and these things. But carboxysomes store enzymes. They store enzymes, and there is a machinery factory that that if you put the carbon dioxide, the thing that will come out will be something like glucose or something organic, right? Organic material. So bisco, of course, is a critical enzyme for carbon dioxide fixation, and that is present in that. The other thing the carboxysome do, I don't think it is written here, that they what was I saying? Yeah, they take bicarbonate and they convert into carbon dioxide, of course, because that's kind of respiration that they do. Now, look at this diagram. This is a bacteria. Forget about these L1, L2. I mean, they're just saying that the outer membrane is protruding inward. These are kind of structures that are present and visible. And if you look at, they have multi-layers, right? And that is L3 layer and L4 layer. Well, there was L4 layer, right? Like these are just layers to forget about. Our work is with these P, P, and PB. Now, what do you think is this PP and PB is? Do we know yet? What? Yeah, polyphosphate. I completely forgot that we have polyphosphate granules. Yeah, guys, we have polyphosphate granules. We do store phosphates as well. So these are polyphosphates, and which is this one? Polyhedral body. Yeah, polyhedral body is. So some has the polyphosphate materials, while PB has the cyanophycin material, the one we were talking about. So, and it looks like polyhedral structure. Like, I mean, the diagram doesn't show it clearly, but you know what polyhedral shapes look like, right? That the hexagon and pentagon and prism and like structures like that. Now, the last one, not the last one, the second last one. Yeah, here they talked about the phosphate granules. Well, they have to talk it here, man. They should do it. Okay, so they didn't talk it here because it is an inorganic inclusion body, of course. Okay, we will come towards it later. So the gas vacuoles, as I told you, they store gases, whatever gas that is. They are being surrounded, and of course, like I said, they use it to float and stuff, right? I don't think I should explain it. A gas vacuole have gas vesicles as well, and of course their walls are made up of protein, but the protein is very simple. It is not anything complex, because of course they don't need it to be complex. No one's attacking it. Now, there are, just as there were important or common types of organic inclusion bodies, there are common types of inorganic inclusion bodies that are present in a lot of bacteria. So the one is the polyphosphate granules. Guys, phosphate is very important because it is present in your DNA, in your nucleic acids. not mine, I mean prokaryotic cells. And there are bacteria or microorganisms that use phosphate as energy source, right? They are known as volatility granules as well. So this is another storage inclusion body that store phosphate. Now they are also known as metachromatic granules, because why? They appear red or different shade of blue. Chromatic means colorful, okay? Colorful. So they appear colorful. Like if you look at the last one, what are they looking like? Black holes, white holes. I mean, yeah, those two colors can you only see. While this one, they appear colorful when you dye it with color. What kind of color? Methylene blue or toluidine blue, right? Now the other type is the sulfur granules. We talked about the phosphate, now we talk about the sulfur granules. And of course they are important for photosynthesis. The more they are independent, the more things they will have in their cells. Understand? Now the sulfur bacteria, they act as the electron donor, and you know how important that is in the photosynthesis and respiration. So in photosynthesis they kind of use sulfur as electron donor, just as we use hydrogen as electron donor in our electron transport chain. It's not the same thing, because in my case we were talking about respiration, but in this case they are talking about the photosynthesis. Anyway, they can be either in the periplasmic spaces, or they can be in globules, right? Like they can be locked up, or they can be present like randomly. Now there is an interesting inclusion body that is magnetosome. As you can see, the name magnet of some is just another inclusion body, right? Magnetosome. It is interesting because it reacts to magnetic field. And why is the need of magnetosome have to be in the bacteria? Is that bacteria magnetic field indicate a lot of things, right? So when there are some bacteria that orient themselves in a magnetic field, right? Like maybe it's because of the quantity, like there are elements that radiate magnetic field, right? So maybe the elements they will need will radiate the magnetic field. They will be there of their use, right? So now the diagram shows that the inclusion magnetosomes are usually linked together to one another, forming chains, right? Like in this case, look at this. This is a microscopic picture. And they are shown. If they are present in a bacteria, they look something like this. These are just normal things. Cell membrane. What was this again? Other structures. Outer membrane. Periplasmic space. Don't concern yourself with them. So if you look at it, it's present this way, and it will be forcing the bacteria to go on to that side. Like in northern hemispheres, it will force it to go towards the northern hemisphere and downwards, while in the southern hemisphere it will lead them to move towards the southern hemisphere and downwards. Now if you look at this, how the bacteria are reacting to the magnetic field, look at them. They made a wave and they are moving towards that magnetic field. So they do react strongly. Like this is not, the magnetosomes are not unique to the bacteria, but we didn't know that the bacteria or prokaryotes can also have magnetosome, because we have seen these magnetosomes in the dolphins. Wait, where are written? Yeah, here. Head of the birds, tuna, dolphin, green turtles. Like how do they know their way? How do they navigate towards this whole world? So they do have things like that, but we didn't know that prokaryotes have the ability to have them as well. Prokaryotes actually, I think they have everything. So this was all about the cytoskeleton inclusion bodies, guys. Don't think that these are all of the inclusion bodies. Inclusion bodies are a lot in bacteria. A lot. I'm saying a lot. Whatever you want to store in bacteria, you have to go towards inclusion bodies. Inclusion bodies are nothing special, right? Like, it's not something complex that you have to work, that you have to need the, I mean, you do need DNA, but it's not like it will be long codes for you to make them. So bacteria can easily make them and remove them, right? And whatever they want to store, they do store it. I'll just be reading the things that I read, and then we will be done. Let's see. Let's read it. Cytoplasmic matrix is a substance in which nucleoid, ribosomes, and inclusion bodies are suspended. We know it is suspended. It lacks organelles bound by lipid bilayer, often called unit membranes. Guys, this unit membrane is a membrane that is for units, right? And in here we meant unit as organelles, right? And of course lipid bilayer. Usually the organelles have this lipid bilayer, the same as plasma membrane. Bacteria don't create something different for the organelle specifically. Not bacteria, I mean eukaryotic cells. It is largely water. About 70% of the bacterial mass is water. Until recently, it was thought to lack the cytoskeleton. The plasma membrane and everything within is called as protoplast. So the cytoplasmic matrix is a major part of the protoplast. So we know that the plasma membrane plus everything within the plasma membrane is the protoplast. So, but the cytoplasmic matrix is not that. The cytoplasmic matrix is just a portion of protoplast. It is just that liquid portion inside the plasma membrane that have these organelles in them. So that's that. The prokaryotic cytoskeleton. Here it is not that much in detail. When examined with the electron microscope, of course they are very tiny, guys. The cytoplasmic matrix of the prokaryote is packed with the ribosomes. So first, when you see the cytoplasmic matrix, it has a lot of ribosomes because ribosome, of course, make its structure, function, protein, secretion, signals, everything is with proteins. And to make proteins, you need these ribosomes. To work, to decode the DNA into proteins, you need that ribosome. So, of course, ribosomes is essential. If you don't have ribosome, you have nothing. You don't live. You die. For many years, it was thought that the prokaryotes lack high level of cytoplasmic organizations present in a eukaryotic cell because they lacked a cytoskeleton. Right? They thought that the cytoskeleton was not present, and by their recent, I mean they are talking about 2002 or something. The book is very old, so it is not that recent in my age, but it was recent. I mean, 2 decades maybe passed. Recently, homologs of all three eukaryotic skeletal elements, microfilament, intermediate filaments, and microtubule, have been identified in bacteria, and one has been identified in archaea. So archaea still haven't been identified with the cytoskeleton. It has been just one. While the bacteria has all of these three, just as I told you, FTS, ZMR, chrysanthemum. Now, the cytoskeletal filaments of the prokaryotics are structurally similar to their eukaryotic counterparts. Structurally, they are similar, and functionally, they are similar, but only because they are present in different cells, so they have to be a little bit different. So that's why the name changes as well, and just to differentiate them. They participate, and of course they are a little bit less complex than eukaryotic cells. They participate in cell division, localize protein to certain sites in cells, determine cell shape. They do a lot of work. Number one is they localize protein, means they transport proteins here and there in the cells to make it available, right? That's one property. The other property is cell division. The other property is cell shape. So here they are showing that FTSD function is cell division. The ring that I showed you, remember? Yeah. So they are, of course, both bacteria and archaea because they both do cell division. Cell division, I mean, okay. So the one cytoskeleton protein that we're talking about in archaea is the tubulin. No, sorry, not tubulin. FTSZ. Remember, FTSZ, not tubulin. Tubulin is for eukaryotes. Now, MREB is for cell shape, and it is only mostly present in rod shape, and very less likely in the circular one, spherical one, because it is cylindrical, helical in shape, so it only can retain rod shape. Chrysanthemum, cell shape, again discovered in Chlorobacter chrysanthus. That's why its name is chrysanthemum. Now, well, you see this diagram, skeleton, this is for the MREB like molecule. Inclusion body. Inclusion body is granules of the organic and inorganic material that often are clearly visible in the light microscope are present in the cytoplasmic matrix. Unlike the cytoskeleton that is only being seen under the electron microscope, inclusion bodies are clearly visible under the light microscope. They are not even talking about the compound microscope, they are talking about the light microscope. Get it? They are large. These bodies usually are used for storage, can be anything, carbon compound, inorganic substances, energy, and are used to reduce the osmotic pressure by tying up molecule in particulate form, right? Like if you have a lot of solute concentration, you will have to bring in a lot of water, right, inside. So to reduce that, to get rid of that, they hide it kind of. They make it a particulate form, they condense it in an area where they think that, yeah, they don't need more solvent for them to dissolve, so it kind of reduces the osmotic pressure. Some inclusion bodies lie free in the cytoplasm, for example, polyphosphate granules, cyanophysin granules, or some glycogen molecules. Like, yeah, these kind of, we will discuss it, explain it. Other inclusion bodies are enclosed by a shell about 2 to 4 nanometers thick, which is a single layer, and many consist of proteins or membrane structure composed of proteins and phospholipids. So they are saying that there are other inclusion bodies that are surrounded by a shell. Inclusion bodies are usually membranous bodies, right? Like they do have membrane, but there are certain that have even a shell around it, and it has proteins in it, or they are like membranous structures. They are structures. Either the proteins are directly present in those inclusion bodies, or they are attached to the membranous structures. And of course, phospholipids can be present as well. Example of inclusion bodies are poly-beta hydroxybutyrate granules, some glycogen and sulfur granules, carboxysomes, and gas molecules all have shells. Many inclusion bodies are used for storage. Many, storage purpose is the first. Their quantity will vary with the nutritional status of the cell. That means if you need the nutrient more and the environment has less of it, so you will want to store it more, so the inclusion body will get better automatically. So it depends on the environment. Prokaryotes react to environment very well. For example, phosphate granules will be depleted in freshwater habitats that are phosphate limited. Yeah, they are talking about, for example, there is a phosphate bacteria that needs phosphate, but the freshwater will be depleted from the phosphate, so they have inclusion bodies for that. Now let's go towards the specificity and explanation. Organic inclusion bodies usually contain either glycogen or poly-beta hydroxycanoids. Like it doesn't have to be butyrate, it can be methanate or whatever. Glycogen is a polymer of glucose unit composed of long chains formed by alpha 1 to 4 glycosidic bonds and branching chain connected to them by alpha 1 to 6 glycosidic bonds. So that just explained what the glycogen is. It is a polymer that have linear chains and branch chains, and of course they have glycosidic bonds. Poly-beta hydroxybutyrate contains p-hydroxy butyrate molecule joined by ester bond. I already explained what ester bond is. So it is formed between carboxyl, COOOH, and hydroxyl OH group of the adjacent molecules. They bond together and the water leaves, and the remaining thing is the ester bonds. Usually only one of these molecules is found in a species, but some photosynthetic bacteria have both glycogen and PHB. Guys, like I said, they both are used as energy source. Like, so if you have one, you don't need another one. But there are bacteria that have both of them. It's not usual to have both of them, but that can happen. Poly-beta hydroxybutyrate accumulates in a distinct bodies around 0.2 to 0.7 micrometer in diameter. They are readily stained with Sudan black for light microscopy and seen as empty holes in the electron microscope. Well, it is Sudan black, so what are you going to see? Of course, you are going to see empty holes. No, wait. I mean, you are not, empty hole means you are going to see white holes, like there is nothing here. It's because they are dissolved in the cell. This is because the solvent used to prepare specimen for the electron microscopy dissolves these hydrophobic inclusion bodies. Glycogen is dispersed even more evenly through the matrix as a small molecule. Like you wanted the Sudan black to make a circular shape or whatever shape of the inclusion body, but it happens indirectly. It stains the granules, and the granule scatters all over the body, making the prokaryotic cell black, while the areas of the inclusion body empty. Get it? Glycogen is dispersed. Oh wait, I have already read it. About 20 to 100 nanometer, guys. Guys, this picture is 100 to 20 to 100 nanometer in diameter. Are we aliens? Aliens or something? We are seeing so inside. If cells contain a large amount of glycogen, staining with iodine solution will turn them reddish brown. Okay, reddish brown. So you can stain them another color as well. Like if you want to see the glycogen, the first one was for the Sudan black is for poly beta hydroxy one, and the second one, the reddish brown color iodine solution, you can say, for the glycogen. Glycogen and PHB inclusion bodies are carbon storage reservoirs providing materials for energy and biosynthesis. Many bacteria also store carbon as lipid droplets. I mean, of course, you guys will know that carbon is not only in the form of glucose, right? Carbon is the backbone of all of the three essential organic molecules that we use. So lipid is one of that. So I mean, the carbon present in the lipid is the lipid droplets, right? If you make it in inclusion body. Now, cyanobacteria, group of photosynthetic bacteria, have two distinct organic inclusion bodies. So, cyanobacteria, they are just, for now, we're talking about generally inclusion body. Now they are targeting a specific type of bacteria. For example, cyanobacteria, they are photosynthetic. So they have cyanophysin granule and carbosome. Cyanophysin granules are composed of large polypeptides containing approximately equal amount of amino acid, arginine, and aspartic acid. The granules often are large enough to be visible in the light microscope and store extra nitrogen for bacteria. As you know, amino acids have a lot of nitrogen in them. So carbosome are present in many cyanobacteria and other carbon dioxide fixing bacteria. Right. Because oxysomes deal with the carbon dioxide, carbon, you can say. They are polyhedral, about 100 nanometer in diameter, and contain the enzyme ribulose 1,5-bisphosphate carboxylase, called as Rubisco in short. Now, why do they have this enzyme? Of course, because of carbon dioxide fixation. Rubisco is the critical enzyme for carbon dioxide fixation, the process of converting carbon dioxide from the atmosphere into the sugar. The enzyme assumes a paracrystalline arrangement in the carboxysome. Paracrystallization. Half. You know half. Not completely crystalline, but yeah, near to crystallization. Like not perfect crystal, but close to crystal, which serves as reserve of the enzymes. Carboxysome also the site of carbon dioxide fixation. They already said it is a site of carbon dioxide fixation. I mean, they can deal with the bicarbonates as well and change them into carbon dioxide. Do I have to tell them what these are? I already told you. Yeah. Guys, for the diagram explanation, go towards the star. I explained diagrams in the start. I mean, I don't want to explain it more. A most remarkable organic inclusion body is a gas vacuole, a structure that provides the buoyancy to some aquatic prokaryotes. Buoyancy, wait, am I pronouncing it correct? Buoyancy? Am I correct? Whatever, we know. It is a floating property, floating. So the prokaryotes that are present in aquatic environment, they do need that. So in many photosynthetic bacteria, photosynthetic because they use carbon dioxide from the atmosphere, so of course they want some mechanism to store them. So you get why the aquatic one and the photosynthetic will have the gas vacuoles, because they deal with the one deals with oxygen more, that's why, and the one needs floating property, and the gas can provide that, so that one has that as well. Now the prokaryotes have example that has halobacterium. Halo, the name suggests that it is a salt bacteria, salt-loving bacteria. Who, whatever halo comes with it is a salt-loving bacteria. Now, Theothrix, a filamentous bacteria. Those both have the gas vacuoles. Gas vacuoles are the aggregates of the enormous number of small, hollow, cylindrical structure. Gas vacuoles. Guys, if you think about gas vesicle, it won't be round. It won't be round like you were thinking about the other inclusion body. Gas vacuoles are irregular in shape. Irregular. They can be small, hollow, cylindrical, and arranged in some stupid ways to make it a vesicle, right? Cluster of vesicles, kind of. Look at this diagram. Do you see any regular shape in this? But it has emptiness in that, and of course this is freezed electron microscopy. It is cutted, so you can see the cutted section. I mean the inside, right? So it is quite irregular. It has stupid shapes. Like, look at this. This is circular, so this is pointy, so this is rod shape, and this is round. Curved. And look at this one. Like, what is this? What is this? What is this? Like, it has complex structure. You don't have to think that they are round because it is gas. So, what was I saying? They descend by simply collapsing the vesicle and float upwards when new ones are constructed. Yeah, wait. So they are impermeable to water, but freely permeable to atmospheric gas. As you can suggest, it is a gas vacuole, so it only allows gas to enter. Prokaryotes with gas vacuole can regulate their buoyancy to float at the depth necessary for the proper light intensity and oxygen concentration and nutrient level. They float towards an area where everything is in their favor. They descend by simply collapsing vesicle. They collapse vesicles mean they pop out the vesicle. They get rid of the vesicle, and so they get heavy and they descend in the depth of the water or aquatic environment. And when they want to float upwards, the new ones are constructed. Two major types of inorganic inclusion bodies are seen in prokaryotes: polyphosphate granules and sulfur granules. Polyphosphate, of course, the chain of phosphate. Sulfur, a lot of sulfur. Nothing to explain. Many bacteria store phosphate as polyphosphate granules or volatin granules. Guys, in the end I will search what is this volatin. It's bugging in my mind. Polyphosphate is a linear polymer of organo or orthophosphate joined by ester bonds. Guys, look, the phosphate has a lot of structures. This is the phosphate, three types of inorganic phosphate. I searched it. So the one is orthophosphate, one is polyphosphate, and one is metaphosphate. If you look at this one, the metaphosphate, this is complex structure, and it has ortho-meta-para position, right? Ortho-meta-para position, or para or meta. I forgot which one was which. So that's why they are metaphosphate, because on meta position they have the bondings, double bond O, right? Now the other one is polyphosphate. They are simple, just simple phosphates combined with one another, while orthophosphate is a single unit, right? And it has double bond O attached, just as this one. But so this is the condensed phosphates. Like, if you condense them in one area, they make it orthophosphates. Wait, am I saying something wrong? Double bond H. So how is it different again? Pyrophosphate OOHO. Okay. Orthophosphate is singular. Okay, singular. It doesn't have it doesn't be in a chain. So what was I saying? Yeah, so polyphosphate is a linear polymer of the orthophosphates joined. Orthophosphate is a single unit of phosphate. When they combine, they make the polyphosphate. And of course, the bonding is ester bonding. I mean, I don't know why the of course came from. Thus, the volatin granules function as the storage reservoir for the phosphate, an important compound of the cell constituents. For example, nucleic acid and what am I doing? What am I doing? What am I doing? Oh my god, so much noise. Hey, close the door. Hey, close the door. Sorry for the trouble. The child is crying. I mean, its cat is, I don't know what happened to its cat. Oh my god, so much noise. So, anyway, who cares about the noise? In some cells, they act as energy reserve and, oh my gosh. And polyphosphate can serve as an energy source in reactions. These granules are sometimes called, oh my god, this is making me angry. Well, let me get them shut up. They don't have to make trouble for me anyway. Right. Right. So these granules are sometimes called as metachromatic granules because they show the metachromatic effect, that is, they appear red or a different shade of blue when stained with the blue dyes, methylene blue or toluene blue. Sulfur granules are used by some prokaryotes to store the sulfur temporarily. For example, photosynthetic bacteria can use hydrogen sulfide as photosynthetic electron donor and accumulate the resulting sulfur in either the periplasmic space or in the special cytoplasmic globules. Do I have to explain this? I already explained it. Inorganic inclusion bodies can be used for the purpose other than storage. Okay? It has more functions than storage. I mean, of course, the enzymatic factory. That's also not storage purposes. But they do store enzymes in that case. An excellent example is the magnetosomes, which is used by some bacteria to orient into the Earth's field. Many of these inclusion bodies contain iron in the form of magnetic field. Iron in the form of magnetite. So here is the further explanation for that. Living magnets. Bacteria can respond to environmental factors other than chemicals. A fascinating example is that of aquatic magnetotactic bacteria. Magnetotactic means they move because of the magnetic field and they orient themselves into the Earth's magnetic field. Guys, remember, they react to the Earth's magnetic field, okay? By Earth's mean, of course, Earth has elements in them. Most of the bacteria have intracellular chains of magnetite, Fe3O4, particles that are called as magnetosomes. Now you get it, what the magnetosomes are made up of? They are forms of, usually, most of them, formed from the magnetite. Magnetosomes are around 35 to 125 nanometer in diameter and are bound by a lipid bilayer. So they are small as well. Some species from the sulfide habitats have magnetosomes containing greigite and pyrite. So if you have S attached to them instead of O, it became greigite instead of magnetite. And if you have sulfur 2 attached to them and one, the chemical formula totally changed. So it is pyrite. Since each iron particle is tiny magnet, we know that the iron has the magnetic nature, right? So it reacts to the magnetic field. The northern hemisphere bacteria use their magnetosome chain to determine the northward and downward direction and swim down to nutrient-rich sediments or locate optimum depth in the freshwater and marine habitats. Again, they react to the elements that they want because they are magnetic and because the elements they want may be present in a magnetic field, strong magnetic field, or they will radiate the strong magnetic field themselves. Magnetotactic in the southern hemisphere generally orient southwards and downwards with a smaller same result exactly. Magnetosomes also are present in the heads of the birds, tuna, dolphins, green turtles, and and other animals, presumably to aid navigation. For helping them in the navigation. What is happening? I'm tired. Animals and bacteria share more in common behaviorally than previously imagined. So we usually think of prokaryotes as something that, they are very different from us. Actually, they are not that much different. They have a resemblance to everything. So here you see I have shown the diagrams, them reacting to the magnetic field, how the magnetite looks like, or how the not magnetite, magnosomes look like, and how they are in chain. So this was it. I hope no one has any more questions. I mean, of course, there is no end to questions, right? So this was it. It was a long day. I didn't know that today's topic would be so heavy, but I hope that you learned something. So goodbye. Goodbye, good luck. Study well, learn well.